Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below.
In the second part of the female reproductive histology, we'll discuss the detailed organization of the uterus, the cyclical changes in the endometrium, the organization of the cervix, vagina, and the mammary glands. The uterus is a complex pelvic organ with numerous structures that are specialized for the gestation and the growth of embryo and fetus. It has two uterine tubes that are specialized for transporting the oocytes and/or conceptus into the uterine body. And each uterine tube, also known as the fallopian tube, has four subregions with distinct histological and anatomical morphology based on their functions. So the distalmost region of the uterine tube is called the infundibulum, which has these finger-like projections called the fimbriae that drape over the ovarian surface. So we can imagine that the infundibulum together with the fimbriae play an important role in drawing in or capturing the oocyte that has been ovulated into the peritoneal cavity. The next region is the ampulla. This is the funnel-shaped and perhaps the longest region of the fallopian tube or the uterine tube. This is where typically the most fertilization would occur between the spermatozoa and the oocyte. The ampulla then continues on to this narrowed and more muscular segment of the uterine tube called the isthmus. Then as the isthmus enters the uterine body wall, the muscular layer of the isthmus blends in and becomes continuous with the muscular layer of the uterus itself. But the lumen of the uterine tube will continue through and traverse the muscular layer of the uterine body as the intramural component of the uterine tube, which eventually opens into this uterine cavity. The main uterus itself has three subregions as well. It has the dome-shaped region called the fundus. It has the funnel-shaped body of the uterus, and then the narrowing at the inferior end of the uterus called the cervix down here. The cervix itself has a narrowed channel that allows the communication between the vaginal canal and the uterine cavity. This channel is called the cervical canal. This internal opening of the cervical canal into the uterine cavity is called the internal os or internal opening, and the cervical canal will continue down and open into the vaginal canal at the external opening or the external os. And lastly, as we can observe, there's this portion of the cervix that is protruding out into the vaginal canal. And this region of the cervix is called the ectocervix. And this is the part of the anatomy that is typically swabbed during pap smear exam to sample some of the cells to monitor the shape and health of the tissues at the cervix. Looking at the individual portions of the uterus histologically, the uterine tube has the mucosa that lines the lumen with a ciliated, simple columnar epithelium forming the lining epithelium and a really thin layer of loose connective tissue comprising the lamina propria. So we can see this higher magnification image of the simple columnar ciliated epithelium with lovely cilia shown here, and underneath this loose connective tissue with vasculature forming the lamina propria. Now, while there are no glands in the lamina propria, the mucosa itself makes many and elaborate infoldings that project out into the lumen. And we can see how this may actually increase the luminal surface area for potentially coming in contact with the oocyte or the conceptus that's floating within the lumen. So the cilia of these lining epithelium play a role in beating in one direction towards the uterine cavity. cavity, thereby generating a current in the uterine fluids to assist in the movement of the oocyte as well as the conceptus. Outside of the mucosa, there's a thin layer of smooth muscles that can contract in a coordinated sequence to essentially generate peristaltic movement, thereby once again assisting in drawing the oocyte and or conceptus towards the uterine cavity as well. So at a higher magnification, we can appreciate this wall that is comprised of kind of loosely arranged smooth muscle layer. And even outside of the muscularis layer, there's a pelvic serous membrane that drapes over the uterine tubes. So we have the serosa. As you can imagine, the histology of the uterine tube varies depending on the region of the tube because they function slightly differently. So the infundibulum with the fimbriae has the mucosa on the exterior of each one of these projections that drape over the ovaries. So we can see the lining epithelium on the exterior and the lamina propria actually forming the core of these fimbriae. The ampulla, on the other hand, has elaborate mucosal infoldings within the lumen with a relatively thin, smooth muscle layer on the outside. That's again draped over by a really delicate serosal layer. The isthmus, in contrast, has relatively small lumen. It does have numerous infoldings, but not quite as elaborate as in the ampulla, but instead the muscular layer, relatively speaking, becomes much thicker. So as always, function, location dictates histology or vice versa. Looking at the uterine histology, similar to the uterine tubes, the main uterus itself is also comprised of 3 distinct histological layers. The innermost layer that's lining the uterine cavity is called the endometrium. So this is the layer. And at higher magnification, we'll see that the lining epithelium of the endometrium is comprised of the simple columnar epithelium of the non-ciliated type. And this lining epithelium is supported underneath by the lamina propria, the loose connective tissue layer. But we'll see that the lamina propria, this loose connective tissue, is actually rather unique in that this uterine stroma, the loose connective tissue, is quite cellular. And many of these endometrial stromal cells respond to the estrogen and progesterone, and they can become quite large and polygonal in a process called the decidual reaction. And the purpose of these larger stromal cells is that if and when a conceptus arrives and starts to implant, these stromal cells can be kind of eaten up, if you will, by the placenta cells and provide nutrients to the implanting conceptus. Now, in addition to these uterine stroma and the stromal cells that can become deciduous cells, the lamina propria also hosts these elaborate uterine glands. A lot of these structures with large coily lumen are the uterine glands. What's unique about these uterine glands is the fact that they too respond to the progesterone and estrogen, so their morphology and function during each menstrual cycle can vary dramatically. So we'll look at this in a little more detail in the subsequent slides. This endometrial layer itself can also be subdivided into 2 regions, the stratum functionale, or the functional layer, and the stratum basale, or the basal layer. The stratum functionale is luminal layer that can undergo the cyclical changes readily. So this is the layer that responds quite well to the ovarian hormones, the estrogen and progesterone. And this is also the layer that sloughs off at menstruation. The stratum basale, or the basal layer, is the thinner and deeper layer that contain the stem cells of the uterine glands. So this layer actually does not thicken or shed in response to the ovarian hormones. So they're minimally responsive to the ovarian hormones. And what's interesting about the uterus as a whole is that the endometrial tissues in the fundus and the body tend to have a pretty plush stratum functionale. However, as the endometrium gets closer and closer to the cervical region down here, the inferior portion of the endometrium has mostly the stratum basale rather than the functionale. So the ideal location for any conceptus to implant and grow would be in the endometrium of the fundus or even the body. The basal or the inferior portion is not the ideal location due to the fact that the functionale layer is actually almost non-existent down here. Now, outside of the endometrium, we have the thickest layer of the uterus called the myometrium. So this is a thick layer of smooth muscles that are arranged in multiple directions. So during pregnancy, this layer of smooth muscles not only undergoes hyperplasia but also hypertrophy in order to support the rapidly growing embryo and the fetus and to generate a sufficient contractile force during labor. The outermost layer of the uterus is the perimetrium. In fact, this myometrium is so thick, we actually did not catch the perimetrium that covers the outside of the uterus. At any rate, this structure, this layer, is the outermost layer of connective tissue, adventitia, or if the portion of the uterus happens to bulge into the peritoneal space, then that region of the uterus would then be covered by the serosa. So the portion that has the serosa would be the fundus and a part of the body, but the rest of the uterus, including the cervix, would be supported by the adventitia on the outermost layer. Now, with the uterine histology, correlating the cyclic changes that occur within the endometrium with the ovarian hormonal cycle is quite important. So we'll look at that aspect of endometrial histology in the next few minutes here. Now, supposing that an average ovarian cycle and therefore the menstrual cycle spanning approximately 28 days or so, with the first day of the menstrual cycle marked by the first day of menstruation, the proliferative phase of the endometrium is the period of time where the stratum functionale is kind of rebuilding or starting to thicken again. So this phase spans approximately day 5 to about 14 in the menstrual or the ovarian cycle. And again, this phase of endometrial histology is marked by the reestablishment of the lining epithelium, which had sloughed off during menstruation, and the lengthening of the uterine glands. So histologically, the endometrium itself will still kind of remain relatively thin, with the stratum basale kind of remaining consistent, but the stratum functionale getting thicker by the day between days 5 to 14. This phase is also characterized by the lengthening uterine glands or the endometrial glands, and these glands are relatively straight and short, if you will. So when we look at the glandular epithelium or glandular morphology, they stay relatively thin and straight. The next phase in endometrial histology is the early secretory phase. So this is day 14 to 21 in ovarian cycle, where there's been an LH surge at approximately day 14, and as a result, the ovulation has occurred and the corpus luteum has now formed. And with the corpus luteum now secreting progesterone in addition to estrogen, the endometrial glands will start to not only continue to grow and lengthen, but also it'll start to secrete nutritious endometrial glandular secretions, which is glycogen-rich fluid. So histologically, we'll start to see the endometrium itself continue to thicken. So I would say that this is about the stratum basale layer and then stratum functionale becoming thicker by the day. And when we look at the glandular histology, the glands that used to be straight and short will in fact start to even coil or spiral. So that's the histology that we're seeing. In fact, the lumen will also start to enlarge a little bit as well. At higher magnification, when these coiled or spirally glands show that they're lined by the simple columnar epithelial cells that are secretory, and in fact, we may be able to see some subnuclear clearing or pale staining regions due to these secretory cells accumulate glycogen within their cytoplasm. So that's the unique early secretory phase histology of the endometrial glands. The latter half of the secretory phase, spanning days 21 to about 28, is called the late secretory phase. And at this stage, histology is marked by the thickest state of the endometrium. So the endometrium here with stratum basale remaining fairly consistent and thin, but relatively speaking, the stratum functionale has really dramatically thickened. Furthermore, the coiled glands will elaborate even further and start to exhibit kind of serrated appearance in the luminal aspect. At higher magnification, you can see that the lumen of these secretory glands have dilated quite a bit, and that lining epithelium is actually protruding in and folding inward into the lumen as well, as these cells have proliferated and are secreting quite a large amount of glycogen-rich fluids. And in case of successful fertilization that may have occurred in the ampulla of the uterine tube, the conceptus would be arriving into the uterine cavity around day 21-ish and start to even implant. And the plushy endometrium and its heavy glandular secretion can support this implanting conceptus. In the absence of implantation and placenta formation, the corpus luteum in the ovary would degenerate around day 28-ish of the ovarian cycle. So with that, the level of progesterone and estrogen would drop significantly, and that will trigger the constriction of some of these spiral arteries that go into the stratum functionale, and thereby cutting off the vascular supply to the stratum functionale, and that leads to the menstrual phase. The first day of menstruation is day 1 of now ovarian and menstrual cycle. So supposing that the menstruation lasts about 5 days or so on average, the menstrual phase would span between day 1 to about 5 in that cyclical calendar. So during this time, the stratum functionale is essentially shedding, and with that, the lining epithelium would slough off along with the majority of the lamina propria and the glands that were in the stratum functionale. Therefore, the histology of the endometrium at menstrual phase would reveal the disrupted or absent lining epithelium. So there's no lining epithelium that we can see from this histology, and that most of what we see of the endometrium would be the stratum basale. And of course, with menstruation, there would be some free bleeding going on. So seeing blood in this histology or luminal compartment is actually expected. And starting once again from day 5 to about 14, we would go back into the proliferative phase where we are re-establishing the lining epithelium over the endometrium. The glands will start to lengthen and grow in a straight and narrow morphology. And as we get into about day 14 to 21 after ovulation, we would have early secretory phase where the glands will start to kind of coil in a nice smooth outline with the stratum functionale growing thicker. And then day 21 to about 28, we have late secretory phase where the stratum functionale has grown even thicker and the glandular outline is a lot more serrated and dramatic, with the lumen being a lot more larger. And in the absence of fertilization, of course, we go back to the menstrual phase here. So that's the menstrual, ovarian cycles, and the corresponding endometrial histology. The cervix is the inferior and narrowed portion of the uterus, and it is characterized by the lack of endometrium or even myometrium. So if we were to take a histological section of the boxed area and look at it under the microscope, here would be the lumen of the cervical canal, and this would be the internal os, and this being the external os, and this being this protrusion, the ecto cervix, we can see a variety of histology that is quite different than the uterine body and fundic histology. The luminal lining actually makes an abrupt transition that we can see right around here from the macroscopic view. The lining epithelium seems quite delicate and thin, but around this region, we can see an abrupt transition to perhaps something that is a little thicker. I might even guess non-keratinized, stratified, squamous epithelial lining based on the location and function of the cervix, as well as the histological characteristics here. This area of abrupt tissue transition is called the transitional zone. And again, this is where the simple columnar epithelium of the cervical lining changes to the non-keratinized, stratified, squamous epithelium. And again, these epithelial lining makes sense because the cervical canal doesn't typically come in contact with external environment or environmental assaults that the ectocervix might, which is exposed to the vaginal environment. This transitional zone and the ectocervix is typically the area that's swabbed to screen for any dysplasia during the pap smear. The bulk of the cervix, instead of being comprised of the myometrium or the smooth muscle, this is comprised of more of a dense, irregular connective tissue. So this region of the uterus remains fairly inflexible and unchanged during menstrual cycle. So again, this bulk of the cervix is comprised of the dense, irregular connective tissue. While there's no endometrium, the cervix, however, does contain an abundant amount of large mucus-secreting glands, which play an important role in mucus secretion as well as lubricating the largely gland-less vaginal canal. At higher magnification of the boxed area, we can once again see the non-ciliated simple columnar epithelium lining of the cervical canal, and in the lamina propria and deeper connective tissue layer, we can appreciate these large and elaborate and somewhat even dilated mucus secreting glands. And during pregnancy, these mucus secretions are a bit thicker. Therefore, the cervical canal is occupied by a thicker mucus plug, which provides some protective role for the developing fetus in the uterus. And here's a higher magnification image of that transitional zone that is approximately at the area of the external os, where the non-ciliated simple columnar epithelium of the cervical canal transitions almost abruptly to the non-keratinized, stratified, squamous epithelium. And this is an inclusion of that mucus-secreting glands of the cervix. The cervical cancer has been shown to rise around this area of transitional zone. So that's one of the reasons why this area is sampled during pap smear for any dysplastic cells. And also of interesting note, during the reproductively active age, the transitional zone actually gets pushed out into the ectocervical space, so that region is a little more accessible during pap smear. The vagina is a fibromuscular tube that connects the uterine cavity with the external environment. A region of anatomical and functional interest might be the recess around the cervix called the fornix. So we can see these circular recesses that go around the ectocervix from the vaginal aspect, which is called the fornix. And these recesses are typically where the diaphragm, for example, is placed up against as a measure of contraception. The vaginal histology is actually relatively bland, if you will, in that the mucosa is comprised of the non-keratinized, stratified, squamous epithelium, as can be expected based on the location and the function. And this lining epithelium is supported by the lamina propria, which is comprised of the blended loose connective tissue, the dense irregular connective tissue, and some strips of smooth muscles. But what is unique about the vaginal mucosa is the lack of any kind of glands. Instead, the lamina propria is fairly richly vascularized. But other than that, there aren't any descriptive, unique structures in the mucosa of the vagina. Outside of the mucosa, there's once again more ill-defined fibromuscular layer that blends with the adventitia. So no particular glands or histologically unique features are the unique features of the vaginal histology, if you will. Now, although the mammary glands are technically not a part of the reproductive system, it belongs more to the integumentary system, but it is rather closely associated with the reproductive function as well as the reproductive endocrinology as well. It warrants a little closer attention during this lecture. So the mammary glands are compound, tubular, alveolar exocrine glands that are organized into the all too familiar lobules and lobes. And this alveolar might be a new descriptive term referring to the glandular histology because we're more used to the term tubulo-acinar, for example, referring to the mixed type of glands. However, we're using the term alveolar to refer to the fact that some of these spherical glandular units can get quite large and dilated and spherical. The mammary glands are present in both sexes, both in males and in females. However, it's during the puberty, under the influence of sex hormones, the breast tissues in females start to accumulate more adipose connective tissues, as well as the mammary glands that start to branch a little bit and are cued a little bit more to elaborate. Even so, in the absence of pregnancy, during the inactive state, the mammary glands are fairly atrophied in their shape. So histologically, what we see more prominently are these ductal components, with the secretory portions being severely atrophied. So in inactive mammary glandular histology, more prominent ducts are appreciated even at higher MEG that are supported by the immediate loose connective tissue stroma, supported on the exterior by the dense connective tissue, but also admixed in that breast tissue would be some abundant unilocular adipose connective tissues as well. During pregnancy, the mammary glands are in their active phase or active stage. At this point, the active mammary glands are characterized by the secretory units or the alveoli that have undergone some dramatic levels of hyperplasia, forming these elaborate and abundant secretory lobules and lobes that are organized around their larger interlobar or lobular ducts. So active mammary glands are characterized by the hyperplasia of the secretory alveoli and the replacement of adipose connective tissues with the glandular tissues. At higher magnification of the secretory units, once again, we can appreciate these large spherical to oval secretory units that are starting to dilate quite a bit, that are draining into the ductal components out here. And finally, after birth, the lactating mammary glands take on even more dramatic histology in terms of the amount of secretory units that have essentially taken over the breast tissue. So the breast is entirely comprised of the glandular tissues, particularly the secretory units, and there's little to no adipose connective tissues in the vicinity at all. Interestingly, once the mammary glands start to lactate, what we might see is some heterogeneous morphology of these lobules. And by that, I mean that some of these secretory units or lobules seem to be kind of almost atrophied in their morphology compared to, say, this region with large and dilated lumen. So lactating mammary glands, it is typical to see the mixture of heavily active and hyperplastic and hypertrophied mammary glandular lobules, and right next door, an area where the secretory units are appearing almost atrophied or non-functional. And that has to do with some lobules that are actively secreting and lactating, while some are temporarily, if you will, taking a rest.
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.